I'm Vicken Karageozian from Allegro Ophthalmics. I'm the president of the company. We're going to talk about our Luminate program for diabetic macular edema. Oh, here it is. Thank you. We're going to give everybody an update on the stage 2 data that looked at sequential therapy with anti-VEGF as well as combination therapy with anti-VEGF in DME. Just a quick refresher, Luminate is an anti-integrin drug. It's a synthetic RGD class drug that binds to multiple specific integrin inhibitors affecting angiogenesis and inflammation. Luminate is unique and very complementary to anti-VEGF, as you'll see. At a very basic level, it turns off the machinery of angiogenesis, meaning rather than working on the signaling side, it's actually turning off the production of further VEGF and cutting inflammation. It's directly anti-inflammatory on leukocyte migration and anchoring. We've shown 12-week durability in the monotherapy study, and we showed it again in this study. And it's been exquisitely safe with no serious adverse events in the drug-related serious adverse events in either DME study. What's interesting about Luminate is it really resets the retina back to homeostasis. It turns off the machinery of angiogenesis in this analogous way. If a normal eye is always producing a little bit of VEGF and other growth factors and slowly clearing them out and keeping it in balance, as we all know, a diabetic eye is producing growth factors at a very high rate, and you're constantly clearing it out with anti-VEGF drugs. The simplest analogy with this, and the reason that we put a kitchen sink together, is with Luminate, it's almost like you're reaching back into the retina and turning off the source. You're turning off the production. Keep in mind, that means that you're not removing the pre-existing VEGF load in the eye. So a patient who comes into your study with diabetic retinopathy or DME will have an eye full of VEGF, and when you give them Luminate, you'll turn off the production of more and cut down inflammation, but the eye is still full of VEGF, and it slowly works its way out. That's very important. In our stage 1 DME study, our phase 2 study that we released back in October of 2016, we looked first at safety, then we looked at dose ranging, and what we found was in 136 patients, the drug was very safe. We met our primary and secondary endpoints with the 1 milligram dose of Luminate, and we found that it lasted for 12 weeks in just about everybody in monotherapy. And we had strong further visual acuity improvements in chronic anti-VEGF users because you have a new mechanism of action. For stage 2, and this is the first time we're showing this information because the data just came out a couple of weeks ago, we looked at a further 80 patients at 14 sites all in the US. Again, 60% of the population is chronic anti-VEGF users. And we basically had a Avastin control arm, monthly Avastin, versus half or 1 milligram Luminate, either in sequential therapy, meaning clear up the VEGF load and then turn off VEGF production and cut inflammation, or use them together in combination. In any case, whether it's sequential therapy or combination therapy, after 3 doses, you went 12 weeks on nothing, when there's, of course, a rescue criteria. I'm happy to report today for the first time that our sequential therapy 1 milligram arm met its primary endpoint. Actually, the half milligram did as well. What that means basically is the visual acuity in green here was non-inferior between Avastin monthly versus Luminate. You actually were at 7.1 letters for Luminate, and that's 12 weeks off of Luminate. You have a mean improvement of 7.1 letters versus Avastin, which is at 6.7 letters, given monthly. What's very important to point out here is look at the far left and you see the red the red syringe. You only gave 1 Avastin injection to clear up the original VEGF burden in the eye, then you reach into the retina to turn off further VEGF production, and you also cut inflammation. If you look at stage 1 DME versus stage 2 DME, last year's data, 1 milligram monotherapy versus now, Avastin helps clear the initial VEGF out of the eye. Luminate turns off the production of further VEGF and inflammation, and what you see in the green line is the visual acuity improvements that we had last year when the drug was used by itself. It worked very slowly because the eye was working against an eye full of VEGF, the drug I should say. A single Avastin injection, so you can have your 1-2 punch, increases the visual acuity by about 40% at all time points. you actually have non-inferiority between Avastin and Luminate at all time points in the 1 milligram dose. One more interesting thing is that if you look at the patients that were enrolled in this study, many of the patients, about 60% of the patients, have lots of prior anti-VEGF, meaning if you look at the first patient, had 6 prior Lucentis injections before they came into the study and never got better than 2400. When you switch mechanisms with this approach, you got down about 5-6 lines of visual acuity improvement, and you see it again and again, whether you're using Avastin, Lucentis, Eylea, or even steroids. In summary, the conclusions for the Del Mar studies, stage 1 and now stage 2, is that in both cases we met our primary endpoint with 1 milligram. 1 milligram in sequential therapy showed visual acuity gains that were equivalent at all time points to Avastin monotherapy. We had strong improvements in both anti-VEGF responders and treatment-naive subjects, and 12-week durability, and no drug-related SAEs. And for my last slide, where do we look as we go forward with Luminate in DME? DME is our lead project. 1 milligram Luminate now in 2 DME studies, late last year and now, showed BCVA improvements comparable to anti-VEGF, 12-week durability, and the ability to treat anti-VEGF non-responders. Use this way in sequential therapy to clear the original VEGF burden in the eye and then hand it off to turning off more VEGF and cutting inflammation. You have your cake and eat it too. You can clear VEGF from the eye, you can decrease VEGF production, reduce inflammation all at the same time, and we think this is a well-matched mechanism of action for the 50% of the non-responder population in DME. Thank you.